I'm here tonight to tell you about the future of cancer detection. But before we talk about the future, let's talk about the past. 6 years ago, I was diagnosed with leukemia. After a few weeks of testing, we eventually realized that original diagnosis was in fact a false positive. A false positive. I thought to myself, how can this happen? Well, unfortunately, it's not as uncommon as I thought. False positives can occur in up to 10% of screening cases. But then I thought, well, that's not the worst that could happen. The worst would be if I had cancer and I was told that I was healthy. Surely that can't happen. Unfortunately, that's not so. The sensitivity of many of these techniques can be as low as 40%. That means that 6 out of 10 people who have early-stage cancer will walk out of the clinic thinking that they're healthy. 6 out of 10. That is unacceptable. And this experience is what motivated me to pursue research into better early detection technologies. So now on to the present. How is it that we screen for disease? Well, generally speaking, we have some tissue that we'd like to observe. So we shine light on it. Some of this light will get reflected, which we can then use a camera to form an image. Unfortunately, with this approach, we ignore a number of different processes taking place. For example, the light can be absorbed as it passes through the tissue. It can be scattered. We also see that the tissue itself can fluoresce, which means it emits light at different colors. So by ignoring these different processes, we discard a tremendous amount of information about the tissue health. What I do is look at how this property of tissue fluorescence can help us improve our early detection techniques. So what is it that we can learn from tissue fluorescence? Well, the first question we have to ask is, what color does it fluoresce at? And so let's consider, as humans, we can see between violet and red. Fluorescence in the violet comes from different types of cell structures. For example, muscle tissue and connective tissue will fluoresce differently. As cancer progresses, we see that the cell structure becomes increasingly abnormal as the disease proliferates. So by recording the fluorescence in the violet, we get a picture of the cell biology. Now, up in the red, we have a very strong signal from blood and oxygen. And so in this case, as disease progresses, we see that it generates new blood vessels to sustain the growing tumor. Simultaneously, this tumor will get larger and larger, and so the center is further from the blood vessels, causing it to decrease in oxygen. So these two factors contribute to the fluorescence in the red, which tell us about the tissue metabolism. And finally, in the green, we have fluorescence from different types of proteins and amino acids, which tell us about the fundamental tissue chemistry. So what I do is look at how these properties change and use them to come up with a unique identifier for tissue, something that tells us whether it's healthy or diseased, and I call this a spectral fingerprint. So then what this means for the future is that I'm developing instruments to actually record these spectral fingerprints in the clinic, so we can correctly classify whether a patient is healthy or diseased, allowing us to treat the patients who need it, and ultimately leading to a healthier population. Thank you.